Hello to all. My name is Dr. Bridget Callaghan, and I'm an assistant professor of psychology at the University of California, Los Angeles, and director of the Brain and Body Lab. Today, I'm going to be talking to you about how our early life environments influence microbiome structure in youth. To start, I wanted to introduce you to my lovely son, Freddie. Here he is covered in mud, head to toe, and absolutely loving it. It's in his mouth, up his nose. In this moment, in this big, wide, and wonderfully dirty world, Freddie is acting as an ecosystem, a home for the trillions of microbes that are colonizing his body. The fact that we live and develop in a microbial world is truly inescapable. Many of these bacteria are reliant on us to survive, and we, in turn, are who we are, in part because of the effects of these bacteria on our development. Here I'm showing you a very great figure from a paper by Helen Wong from Elaine Shao's lab at UCLA. And what you're looking at is a mouse head with a mouse brain inside, and these colored boxes represent different brain regions. In the boxes associated with those brain regions, you can see the multitude of neural effects that are associated with growing up germ-free. So mice that have no bacteria in their bodies from early in life. So what you'll notice is that the brain develops along a very different trajectory if you grow up germ-free. And this is a very developmental effect because reconstituting the microbiome after development ends, so more in the adult period, actually doesn't help to save many of these brain profiles. You need to reconstitute early in life in order to get the brain that we know and are familiar with. So in other words, the first take-home message today is simply that we need a microbiome in order to build a brain, or at least to build a brain that we're most familiar with. Now, the microbiome is a developmentally regulated organ. Starting even before birth in many cases, the young microbiome community is initiated, and numerous factors shape its growth, including but not limited to genetics and mode of birth, diet, the transition to solid foods, environment, having a pet at home, siblings, stress, as well as medications. In other words, we know that the microbiome features are changed by things that are occurring early in life. And that's not really that surprising when we see how much the microbiome changes early in life. So there's lots of flux, lots of room for environmental influence. So that's demonstrated really nicely here in this figure, showing that the number of bacterial groups, we can think of this as, change enormously, increase enormously across the first 4 years of life and then seem to taper off thereafter. So in other words, what we're seeing is this period of growth or flux in the microbiota, and this is when we're seeing the environment taking playing the biggest role. So what this might look like across development, so if we're putting it kind of on a time frame, is that microbial plasticity, while it is highest around birth and the weaning age and the first few years of life, rather than tapering off entirely, it does seem to be maintained at some level across the lifespan. And there has been some suggestion that there might be another sensitive period or bump in sensitivity in the microbiome to environmental influences in adolescence, but that remains to be determined. That needs to be investigated much more closely. So something that you might notice about these microbial plasticity peaks, if you're a brain person, is that they actually overlap significantly with periods of plasticity in the brain. So we know that brain development is hierarchically organized. We have some basic functions emerging first, followed by a higher order cognition, which kind of builds on top of those functions. So if a plastic period is a time where the environment is more likely to shape or mold a system, it's possible that one event may actually affect both microbiota plasticity as well as brain development. And it's also possible that we might see interactions here. So potentially changes that are going on at the level of the microbiota may affect brain development and vice versa. So we've long known that adversity is associated with a higher incidence of mental illness and an altered pattern of brain function, which underlies mental illness. But understanding this mechanism hasn't really helped that much in terms of treatment because, of course, it's very difficult to intervene at the level of the central nervous system. Our brain is locked inconveniently for neuroscientists safely inside of our skulls. However, if we know that bacteria are disrupted following early life adversity, then this sensitive period framework tells us that we might be able to intervene at the level of the microbiota in order to hopefully influence both brain as well as behavior. So a lot of my work has been focused on investigating this link between adversity and microbiota disturbances in youth. In my work, I've tended to focus on a really extreme form of adversity. So that is children who are raised in institutions or orphanages, such as this one depicted here. These are characterized by extremely high child to caregiver ratios and the lack of a stable and consistent caregiver. It's really one of the most extreme forms of adversity that a child could endure. The children who participated in my studies have been adopted from these institutions, usually by about 2 years of age, and have been living with their adoptive American families for many years at the time of study enrollment. However, we see that even when controlling for a range of covariates, this event seems to have a lasting impact on the microbiota. So here are some early data from the group of early adversity or EA exposed children and a comparison group of children who were always with their biological caregivers. And just to summarize a large study, what we saw in this sample was that adversity was associated with lower bacterial richness, differences in beta diversity, so the adversity samples clustered together and separately from the comparison group, as well as differences in the relative abundance of specific taxa, in this case, Lachnospiraceae. We've since been replicating and extending that study. So we now have in PrEP 2 different studies in different age groups. So one in 0 to 3-year-olds and another in 6 to 18-year-olds. And we're again seeing that adversity is associated with an altered community composition of bacteria. So richness and diversity, or diversity in this older sample, as well as again, the relative abundance of specific taxa. So take-home message 2 then is that early life adversity does seem to impact the developing microbiota. And while there's a lot more that we need to learn on this front, so we need to do much bigger studies and much more intensive sampling, we also need to look at the function of the microbiota, not just the structure. It does tell us that the microbiota may be a potential target that we could look at in terms of affecting behavior and brain health. And so I guess one question is, can we treat changes that occur in terms of microbiota being impacted by early life adversity? Now, I think we're too early to begin looking at this in humans, but we do have some preclinical data in rodents that I'm going to show you now. So here we have rodents who have just been exposed to standard rearing. They've always remained with their caregiver. And you'll see that when they're trained to fear a white noise that's paired with a foot shock, they then show freezing or fear responses to that white noise soon after the event, but by a couple of weeks later, they forget. So this is a phenomenon known as infantile amnesia. In contrast, rats who've been exposed to maternal separation stress, being taken away from the mother, show excellent fear retention, both recent and at remote intervals. However, if we pair maternal separation with a probiotic in the drinking water, we actually see a rescue of this behavior. So these animals begin to again display infant-like behaviors. So probiotics can actually reverse some of the effects of adversity on emotion behaviors. So take-home message 3 is that adversity effects on microbiota might be treatable, and this may have implications for mental health in humans. So to summarize, the microbiota is a really interesting target to look at, particularly when trying to examine the implications of early life adversity in humans. I'm going to end by saying an enormous thank you to my lab, to the students in my lab, and to the families who participate in our research, as well as to my funding sources. And thank you all for listening.